Okay, so
Indonesia Hospital Expo
for the sample collection, so tube is a system with several kind of
Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi, salam sejahtera bagi kita semua. Pada kesempatan kali ini, PT Gandasari akan memperkenalkan produk Copan dari Italia yang akan dibawakan oleh Mr. Cristiano Sabelli di acara BMF Hospex ke-13. Nah, dengan judul Copan Evolution of Sample Collection, Self Collection and Transport, yang akan membahas tentang pengumpulan sampel dari pengumpulan sampel sampai transport yang sampai kini dapat dilakukan dengan mudah. Jadi, selamat menonton.
Okay, so I repeat, thank you for this invitation, and today we will discuss about the Copan products in terms of the evolution of sample collection, self-collection, and transport. So, just to start, why swab? And later on, why Copan? So basically, one of the routine procedure that you had to use in the diagnosis of infection, a critical step is the sample collection, and swabs allow for a safe and effective collection from different body sites, and allow also in combination with transport media to transport the sample from the collection site to the laboratory. However, there are different kind of swabs, both in terms of shaft and tips. So you could have shaft made on wooden, for example, or plastic, and tips made of dacron or nylon. From this point of view, Copan introduced more or less 20 years ago a revolution of these simple but critical step for the sample collection, introducing the Copan FLOQSwabs. It is a device is a sterile and ready for use system for sample collection. The main differences between the past of the swab collection was mainly based on the flock technologies. So the introduction of moving from cotton fiber swabs, so these long fibers of cotton, for example, to the flocked swab. As you can see on the screen, on the left you have a microscope image of a fiber swab, so you can see this kind of web structure for the classical swabs, and on the right you see a microscope image of the flocked swabs. So kind of structure that we call also an open structure, and the differences between these two simple structures are related both to the absorption and to the release of sample. This means that this structure for the classical swab is a sort of can interfere with the absorption of the swab of the sample, while with the flocked swab you have this open structure allowed to absorb almost the entire volume of sample you need to collect. On the other hand, also for the release of the sample, samples with a cotton swab is practically trapped inside the swab, inside the tip, and the release is the 10% in comparison with the release of a flocked swab that can also achieve can be higher than 90%. So you can absorb almost the entire volume of the sample, and you can release almost the entire volume of the sample. So this is basically based on this superior elution of the biological specimen. Differences are also in this case related to the sensitivity you can have with with a FLOQSwab, with a sample collected with a FLOQSwab in comparison with traditional swabs. As you can see in this simple image, you can see for the sample collected with FLOQSwabs an higher number of colonies. So you have an increased sensitivity in terms of number of colonies, but also in terms of how many microorganisms you can identify starting from a sample. To produce the combination of a plastic swab, a plastic shaft, and this flocked tip can also allow to increase the possibility to better collect a sample from different body size. And this is because you can pre-mold the shaft geometry to be to increase the possibility for an effective collection, and with the FLOQSwab you can maintain this geometry also for the tip to increase all these points. As you can see, for example, this is a picture with all with our different FLOQSwab. So different, you can see different geometry for both for the shaft and for the tip. And for example, with this pre-molded plastic shaft, you can have for the nasopharyngeal swab that is really well known also during the pandemic. For example, the presence of a ball at the end of the tip that can increase the sample collected, or you can see also different swabs on the left and on the right for the mid-turbinate collection. So you can shape your swab just to increase all the sample are collected. And these differences in geometry, in shape of tips and shaft are related to the different collection sites that you can achieve for the sample collection. So starting for example from the first one, and on the top you see for example our L-shaped swab that is used for cervical collection, but you can have also the last on this image are also related to the nasopharyngeal collection, both for example in adults and in pediatrics. So this combination can be widely used to optimize the sample collection. And these increase respect than the past for the sample collection and dilution in combination with an LBM, so with our transport medium, can allow to have an effective sample collection and transport and a higher sensitivity. So this open structure of the FLOQSwab increased also the possibility to use our transport medium to have an optimized release, a sample more homogeneous in this liquid-based medium, and of course also to have the possibility to start with the automation, so to save time, increasing the automated part of the lab routine. One of the very interesting application of our FLOQSwabs, also related to the easiness of the collection, is the use of Copan FLOQSwabs also for vaginal self-collection. Practically, this is something that can allow this self-collection procedure is something can allow also to improve the participation to the cervical cancer program involving under-screened or never screened women aged based on the country of the program from 25 to 75 years old, and this allow improve the accessibility for all women to cancer screening program, considering also remote or rural areas. So the first question we usually had when we started with this kind of project with this product, where if is HPV testing with a self-collected vaginal sample can be comparable or accurate with a clinician-collected sample? And the answer is basically yes. So there are up to now there are several already published paper, and we will see also several screening programs that are in this direction. So are demonstrating that you can use with higher sensitivity this possibility to compare results you can obtain from a vaginal collected sample and from professional or staff collected samples. So in this case, you can also to increase this participation, so the women can collect their own sample in a point of care or also at home. And this is at the base of our self-vaginal FLOQSwabs products. So a comfortable device that can support women taking part with the HPV, but also for the STI, for the sexually transmitted infections screening campaign. So we are speaking of 4 different product codes. Basically, the differences are in the presence or the absence of a breaking point, and the breaking point allowed to broke the swab in a tube for an automated or for a better used in the lab. And we have two different for the considering the breaking point. We have 2 different positions for the breaking point. So you can collect samples starting from 80 millimeters or 60 millimeters, and all these swabs were validated and are validated for the HPV or STI detection based on a molecular diagnostic test. This is a rough of our instructions. So these are our instructions. These are the instructions provided to women for the collection. So instruction should be followed to collect this kind of samples. And we defined all the critical aspects to have to increase the to obtain an effective sample collection in all kind of environments. So point of care, where also a clinician can be present or not, and at home, so completely intimate. So the key points of this product is the safety. So several safety aspects. So no, at the moment, no adverse events were registered with this product. Instructions were found very clear and adopted at least for images and basic instruction, also in several pilot studies, and the presence of the red mark to highlight the presence of the breaking point, but also as a sign, as indication on where to put the fingers during the collection, allowed to have a really safe device. And the effectiveness, from this point, we compared the results from the vaginal self-collected samples with the clinician collected samples, and the agreement is very, very high. No invalid were detected during the study, but also now that we have this product also on the market, and we have a high sample stability. So you can have for the 2 classical STIs as the chlamydia and the Neisseria, you can have 4 weeks also at room temperature, and for the HPV virus, you can have 2 weeks starting from -20 Celsius degrees and 50 Celsius degree. So this is the main points. Why this is the reason why several different screening programs are now you are currently using our device, and the compatibility in the laboratory with several kind of platforms. So you can collect the vaginal sample, you can self-collect the vaginal sample, you can elute with classical medium, alcoholic-based medium as the preserved seat, or and this is under validation, currently under validation, and used in some tests also in Australia, for example. You can use also one of our transport media, the Copan swab. In general, all these products are basically compatible. The all samples eluted in these two media are basically compatible with the test with all platforms are currently and widely present in the laboratory, starting from the Roche Cobas to the Abbott system, or the BD Viper, Cfade, and the Cgene assay for HPV and STI. This is a short list of pilot studies or screening programs are currently ongoing or just finished that's involved the use of successfully involved the use of our Copan FLOQSwab for vaginal self-collection. So the next point I would like to discuss is also to present is also our transport medium. As I told you, the possibility to use this compatibility, this high elution characteristics of the FLOQSwab in combination with our LBM, with our liquid-based medium for the liquid-based microbiology, increase also the possibility to have to automatize all the steps in the laboratories, all the routine procedures in the laboratory to have a high efficiency of the lab. So the first one is the Copan UTM, really unfortunately well-known transport medium, well known because was widely used in all countries worldwide for the detection of the SARS-CoV-2 during the pandemic. Is basically a medium that allow to collect and transport clinical specimens containing viruses, chlamydia, mycoplasma, and duraplasma. There are several characteristics that can be mentioned for this product. For example, the presence of 3 glass beads that increase the possibility to homogenize the sample, the compatibility, of course, with the FLOQSwabs, and the presence of kits with different FLOQSwabs with different shape for the shaft and for the tip. Is a medium complying with the CSI M40A2, so the guideline, a US guideline for the reserve of clinical sample up to 48 hours. And can be available under development, and the product will have also a pierceable and resealable cap, and of course is compatible with our automation. And this is one of the last development of this product, is also compatible with the with a pooling strategy using our Copan pod swab, and we will see later for this product. So these are a scheme of the characteristics of this product, considering different geometries, possible fields of application. So starting from the respiratory infection, but also in STI, You can collect samples from different sites, so from nasal, oral, nasopharyngeal, and so on. And there is a wide downstream application that you can apply with sample collecting with the UTM, from the classical culture to nucleic acids testing. And here there is just to give you an example of the wide compatibility of the product with several diagnostics platforms. Widely used, as I told you, during the SARS-CoV-2 pandemic, and this is also what Copan is also doing, is to provide rough collection guide for the to collect different kind of samples. Of course, this is in any case something can be managed considering the internal procedures and national procedures for sample collection. And UTM pod swab is we are speaking on a medium on an UTM tube with 6 ml of medium, but in combination with 5 swabs. 5 swabs that allow to collect 5 different samples from 5 different persons, and so you can increase the you can save time and reduce costing, testing 5 people per time, and you have only to call back people are in a tube where there is a positive sample. And difference is between a classical 3 ml UTM, so single swab in 3 ml UTM, and the pod swab is in terms of diagnostics results, there is a difference in PCR of 1.5 CTs. So really are really comparable. The other one is Copan Italy. Copan Italy is different from Copan UTM simply because there is not it's a transport medium, but mainly dedicated to RNA DNA, so to the nucleic acids. In fact, this is a medium that allows to completely inactivate all the microorganisms are present in the sample in 30 minutes. And the presence of guanidine isothiocyanate allows nucleic acids from degradation, and the stability of the samples that you can achieve with this for the sample is up for compare. So it's really useful when you have to collect a sample from remote areas, and you can transport for the molecular diagnostics with in condition that can need to stabilize the sample, the nucleic acid sample, up to waiting to test the sample in the lab. Also, in this case, the combination with different FLOQSwabs with different geometries allow to collect samples from several collection sites, and you have in this case several fields of application. And we introduced also the microbiome application with this product. And is as I told you, is mainly based for molecular based assay and NGS, so the sequencing of your sample. Compatibility is well certified for all the platform currently mostly used in the laboratory, and we can discuss on a further device, and this is different. This is different from FLOQSwabs. Is a collection device mainly dedicated to the collection of saliva. So why saliva? Simply because we found that the scientific community found that that SARS-CoV-2 can be also detected in saliva samples. and is detectable in symptomatic and asymptomatic patients. The differences with nasopharyngeal swab is, of course, that the saliva collection is less invasive and is compatible with self-collection procedures, both at home or in point of care. And one last is also that you can use these self-collection procedures also with critical population, for example, with children or in longer care term facilities. It can be, of course, these allow also to be more dedicated to healthcare professional. Can be dedicated to vaccination, to other activities than the sample collection. Lollisponge is based on sponges inserted on a plastic shaft, and this shaft is inserted in a screw cap. The tube, this system, sponges and cap, allow to collect the sample really in a safely manner, both for the patient, but also for clinicians. So you don't have contamination on the outside of the tube, and also in the lab is easily used for considering a sanctification step and high comparability of results with the nasopharyngeal, considering that mainly the saliva samples are positive in the first 3 or 5 days from the onset of symptoms, or so in this range, in this time period, you have the highest comparability with the nasopharyngeal swab. Also, instructions are really easy to be used. We are speaking on two steps. So the first one is to collect saliva on the tongue, and one for 10 seconds, and to collect with the Lollisponge this saliva in the mouth for 60 seconds. So really easy to be followed by patients. And so user advantages or user acceptance is really high, of course, in comparison with a nasopharyngeal swab that is more invasive. And these allow to new opportunities. So, with a nasopharyngeal swab that is more invasive, and these allow to new opportunities. So, professionals can be assigned to other tasks, or you can use for mass screenings. For example, in Italy, this product is currently used also for screening in school, where children are tested every 2 weeks, and the device can be used at home without any kind of problem in this home environment, and they can use it also after in the lab for the collection. Of course, this device can be used for respiratory infection, and can be tested with nucleic acids methods, and what is ongoing is also for antigenic testing, so using a rapid antigenic test. One possibility is also for the lab to use the sample collected with the Lollisponge with the crude extraction. So this allows also to skip the phase, extraction phase in the lab, so saving time and also reducing cost. And we tested with several kind of platforms based in basically the most used in Italy. So where the assay where we have more data is the kit from Sigin, but it's also widely used with several home-made test, or for example, also with the CDC test. The test was developed by the CDC at the very beginning of the pandemic. Lollisponge is our product is a similar as a concept is used to collect urine. It's based on sponges, on free sponges present on a plastic shaft, and it allows to absorb urine that can be released with a brief centrifugation step in the lab, and the product can be used to detect urinary tract infection. It's compatible with the automation, and is used for the culture of aerobic and anaerobic microorganisms. A variation of the urine sponge is the self-urine sponge. The device is similar. The differences are in its intended use is dedicated to the collection of HPV virus and other STIs to be detected with molecular assay. In this case, you have a stability of 1 week at room temperature or 3 weeks at 4 Celsius degrees. Is really safe because it doesn't contain any kind of media, so can be used at home in a safely manner, and is compatible with our automation, so with the Copan universe, considering these differences between the urine sponge that is used for culture and wasp, and this self-urine sponge that can be used for molecular diagnosis and together in combination with Copan universe. These are also, in this case, we have the instructions are really clear to be followed, really easy to be followed by the patient. And this is a list of what I already said for the ASCAR. To summarize, the FLOQSwabs increase the possibility to collect and to release samples in transport medium. In particular, the possibility to use the vaginal self-collection FLOQSwabs allow to increase attendance in screening programs, and the possibility to combine the use of FLOQSwabs with Copan transport medium allow to preserve all collected samples, ensuring its stabilization from the collection site to the laboratory. So, and thank you for your attention for these 30 minutes with me. Thank you.
Nah, itu tadi pemaparan video dari Mr. Cristiano Sabelli. Nah, buat teman-teman yang masih bingung karena videonya kepanjangan atau terkendala bahasa, saya akan bantu ringkaskan menjadi 3 poin secara garis besarnya. Poin pertama yaitu Copan FLOQSwab. Ini adalah suatu swab yang digunakan untuk pengambilan dan pengelusian pada sampel biologis. Yang poin kedua yaitu vaginal self collection dengan FLOQSwab. Ini digunakan untuk meningkatkan peminat pada screening HPV dan penyakit menular seksual lainnya. Poin yang ketiga dan sangat penting yaitu Copan Lollisponge. Lollisponge adalah suatu swab yang berbahan dasar spons, satu-satunya di dunia dengan fitur utama 1. Tutup tabung beraroma lemon, jadi memicu kita untuk memproduksi air liur sebanyak-banyaknya. Kedua yaitu tabung kedap, jadi kita tidak perlu khawatir sampel akan tumpah atau pecah. Tuh. Kemudian yang ketiga, tabung bebas cairan, jadi di dalam tabung hanya terdapat spons saja. Nah, gimana? Masih ada yang bingung atau ada pertanyaan? Kalau ada pertanyaan silahkan tinggalkan di kolom komentar dan komen. Kemudian jangan lupa follow IG Gandasari Ekasatya dan Facebook Gandasari Ekasatya. Jangan lupa like, komen, dan subscribe. Terima kasih.